Oh, don't worry about that. I didn't even notice. If you hadn't pointed it out, I'd never notice. Right. So we want to download the internal genes PB1, PB2, PA, and so on from H5N8. So looking at the NCBI interface, I'll just use this one. I could have used the influenza research database. It's pretty much the same. So I don't mind. It can be from any host. It is definitely A, and it can be from any region of the world. The particular proteins that I want are the internal ones. Now, you're going to need to do this one at a time, because you need to make sure when you download sequence files that they're homogeneous. They contain the same stuff, because if they don't, you get in a mess. They want to come from H5, so you pick that in the subtype, and N8, you pick that in the subtype. You pick full length only, and you go, if I missed anything, show results. So this will show me all the PB2s. So there are 268 PB2s, which I can then download. Download this protein faster. Now, remember I said about having customized faster define? Because I'm on the same computer I used last time, I think it's done it fine, which is good. Otherwise it'd be in a mess. So it immediately just saves it to download in a not very useful file name, which is faster.fa. So I'm going to rename that, and remember we have specific ways of renaming it. 2021 25 NCBI PB2, and it's from H5N8. I didn't. H5N8, and I'll put .FAS. Yes, I do want to change it. What? Okay, you like that? Surprised because it should have changed. Icon, doesn't matter. Right, so I've downloaded the first of those segments. Now there's 6 of them. I was just I just double clicked on it to open it in Mega, so I can have a little look at it. Interesting. Yes, potentially. So what you're doing when you look at these things is looking for patterns and looking for regions on the top when there's nothing that is the same. So that tells you there's going to be a bit missing somewhere. So this sequence is not good because it's got a big bit in the middle that's rubbish. So I just delete it. Now to clean up your sequences to start with, you want to look along that top line, because the stars represent things which are the same in every single row of that particular column. And the DNA space, there's a bit more variability than in the protein space. So if I'm looking at a protein sequence like this one, then I shouldn't have big gaps where there's nothing like this. This indicates that there's some kind of problem. So if I scan down the sequences, here's my problem. It's not a very big problem, it's a teeny problem, but anyway, it's a problem. So what's happened here is, if you look at this sequence, the P is in the right place, then the D is in the right place, then this G is not in the right place. It should be next to it in the G and the T. So what I need to do is grab that whole thing and drag it across to make sure that these things are aligned properly. So what I I've also done this week is I changed my simulations to show how sequences get out of alignment and why we need to do this process of alignment, which is what we're going to do next week. But it's quite good that I didn't need to create that false data because I've got a set of data here. So the reason why it all breaks up after this point is because this particular sequence is missing an E from this particular position. So what I can do to sort that out is I can do alignment. Now, muscle is a good aligner. It's the best of the two choices you have within this particular program. So yes, I want to select all of the sequences, go and calculate it for me. It's 268 sequences, which is not a trivial amount. So trivial's 10 or 20. But I'm looking at proteins, so it should, and there's only one serious protein that's any different, which is that one. So what it's done is it's inserted a gap where that particular E is missing in that sequence. And now we've got loads of stars at the top. So that is all good. That is an aligned sequence which is being cleaned up in protein. Now the thing that you should also recognize is, so there's sometimes it's L here and sometimes it's Q here. So all the ones that have Q there are closer relatives than all the ones that have L will form another family, and these are called lineages. So these are the different mutants. Interesting over here that this particular sequence has changed a D to an A twice, and that pattern is not seen in any other sequence. Now, we have a choice of two things here. This is either something interesting happening or it's a cock-up. My tendency in this is to think that this is a cock-up. I think that somebody's messed up the DNA sequencing at these two positions and called the nucleotides wrong, and that the particular codon is close to the D codon, is an alanine codon which is close to a aspartic acid codon, and they've just made a mess of the sequencing. So some changes are reasonable and some aren't. So the charge residue, like an aspartic acid to an alanine, is not really a usual change. You'd expect it to be something simpler. So if you have a column where there's only a couple of sequences changed, so these are two H's here, are there any more H changes? There's another one there. So again, they form into families. But because there's only 3 changes there, this is a more recent mutation than the one that we had further across, which was those L and Q mutations, because they're roughly split, all the sequences half and half to be L and Q, whereas almost everything is H. So that means after L and Q separated from one another and kept evolving, the H arrived later, because it's less common. So working out those evolutionary relationships, here's the LQ. So working out those evolutionary relationships allows you to work out what the ancestor looked like and how evolution has evolved the sequences along the different branches. And this allows us to construct trees. And that's what we're going to do for a lot of the module. Tree construction and alignment. Right, so I've got, that's the first one. If I wanted to get the DNA sequence as well, the coding region, I could just click on coding faster, and then I can download that, and it'll download another file called faster.fa. That's why you keep having to name these things. Oopsie. Right. So I call that 2021-25 NCBI PB2 H5NA. Now what am I going to call this one? Because it's 2021. It is 2, 5. It's still from the NCBI. It is still the PB2. And it's still from H5N8. So stupid me, I should put this to be DNA. Fast. And now I need to go back and edit this one's name to call it .aa.fast. So that tells me I've got 2 copies of exactly the same thing, but one's as DNA and the other one's as amino acid. It's really important when you're doing bioinformatics to be systematic like this, because if not, and you come to doing your analysis and you look at your files and it says seek 1, seek 2, faster 1, faster 2, you'll be sitting there going, I have absolutely no idea what's in each of the files. So that's a mess. So I'm showing you there, I did that correctly. So let's go and do something wrong. So remember when I told you I need to download each of those files distinctly, each of the sequences, because otherwise I'll get a mixture of files. So let's go and do it wrong. If I go, I'll just have to do back. So if I go back to here, so instead of having just PB2, I also include PB1. So now I'm downloading 2 separate sequences at the same time. So now if I show the results, hopefully I get 500. Yes, double the amount of sequences. That's all good. So let's download the protein faster here. See what it is. So it's giving you a warning to say this isn't a good idea because we won't be able to do alignment and clustering. But we've done it anyway, so let's just download it. So I'll download it into faster.fa, and I will not edit the name this time because I'm going to throw it away as soon as I've shown you what happens. So convert it to a fast file. Yes, I'm sure. Now, remember, when we've got everything together and we've done it, no, I don't want to save the current one. So remember what I said, you want to have loads of stars at the top. So we're not getting a load of stars, but it could just be because it's not very well aligned. So let's tell it to do some alignment. Okie dokie, this will take a while because it's 500 sequences. Hopefully it won't take from now until Christmas. It can do, and doing things through Mega are slow. Mega has 2 alignment programs within it, ClustalW and Muscle. Now, I always use Muscle, and I've got fixated into using Muscle and always considered Muscle the best thing. I was the other day looking through the literature as to what's the best alignment program, and they were saying not Muscle. I was going, oh. Now, ClustalW is definitely the worst of the alignment programs that still exists. They had a replacement to ClustalW, which is Clustal Omega. So if you're going to use a Clustal, you use that. And it works about the same sort of efficiency as Muscle. Sometimes it gets better results than Muscle, sometimes it doesn't. There's another program which is called MAFT. And MAFT is much faster than both of them. And oddly enough, MAFT also produces better alignments according to an evaluation than both of them. So you see here, I've got a very nice mosaic kind of pattern. This is what happens when you try to match apples with oranges. So all of the proteins start with an M, which is the methionine. And apart from that, there's not many amino acids that align along the whole length of the sequence. Sporadically, you see some things and some particular patterns. And this is because you're aligning the polymerase B1 gene with the polymerase B2 gene. And you can easily see that there's two very distinct proteins here. So there's the ones which have this LPD and whatever, and then there's the other ones that don't. So you end up with this very partial kind of alignment. From the way it's actually set up, you can tell that in the very distant past, these two proteins did come from a common ancestor, but they've diverged over such a long period of time that they're no longer easily comparable. You can see this because of the patterns of colors. Because you get these blocks which share similar patterns of colors and movement of colors, it's likely that they did in the very distant past share an ancestor. But if you get alignments like that, you've done something wrong. No, I don't want to save that.